And so with that, I would like to introduce Professor Jean-Baptiste Cazier and Professor Ben Brown from the Centre for Computational Biology, who, I joke about Jean-Baptiste, but Jean-Baptiste is the person who's really pushed us on this to deliver the resources to help his research, and it's benefited the whole university. So, Jean-Baptiste?
Thank you, John. Thank you, John, for the introduction, and I'll take the blame happily for this. My job here is just to be introducing Ben Brown. You see the title in a second, so I'll try to be brief. I'm going to, if you can see from the presentation here, is going to talk about linking the genome dynamics to ecosystem function, and underlying you will see the word bioinformatics popping up. So I'm just going to try to do in 2 slides to introduce you what is bioinformatics, or what I think bioinformatics is, and then to see what the university has done with the help of the IT research to help us provide that. And you will have the best example with Ben later on. So bioinformatics, if you haven't heard, BBC earlier this week, who discovered bioinformatics and gave a definition of it. What is it? You can see it is made of bio and informatics, so it's very relevant today because we are in an IT place, IT solution to look at biology. But if you talk to people all across the world, all across the field, they will come up with different definitions. Some will say it's computational biology, some will say it's biostatistics, statistical genetics, mathematical biology. You can come up with a lot of terms. So everybody's got his own definition. So when that happens, you go to the reference. I used to work in Oxford, so I look at the Oxford dictionary, open the big book, see what does it mean. And I looked at it, and there was nothing. It's very disappointing, because this was my new field. I'm a mathematician turned bioinformatician, and my field doesn't exist. Very sad. So I thought, no, we have to be resourceful. We are going to go on to the online version of the Oxford Dictionary, because they all have spires, but they have an online version of the dictionary. And they give a definition, which is pretty good, which says that it's a science of collecting, analyzing complex biological data, such as genetic code. I like pretty much every single word in this sentence, because it's a science, first of all. It's a tool, but it's a scientific tool. The part of what it does is collection of data, so it's database, but as well we do a big part of analysis of the data, and the data is not simple, it is complex, essentially biological, and such as genetic code. So we do a lot of genetics, but we don't do only genetics, we can do a lot of different things. So this is a very good idea of what bioinformatics is. But you know, nowadays, if you want to have a proper definition, you go to Wikipedia. That's where you get the proper things. What does it really mean? And I just took a slice of it, because, of course, it will be very long. And just taking a slice of it, you don't need to read what it says. But I try to put in color the different fields or keywords that are coming up. And in there, it can be algorithm, it can be database, it can be modeling, it can be system theory. Bioinformatics is pretty much all of those things. And if you're geeky enough, you will understand this reference, that bioinformatics is pretty much life, the universe, and everything. We are always there. You can't get away. So with that in mind, this is, you may recognize, this is the University of Birmingham has recognized that very early on. And under the impetus of John Frampton and Kevin Shipman a few years back, the university decided to create a Center for Computational Biology, which is been built in that way. So you may recognize one of the persons there up there. So the idea is to provide a center of expertise in bioinformatics or computational biology across campus, being able to talk with everybody all around. So we have 3 main branches. We have bioinformatics, I've got the chair. You have a chair of clinical bioinformatics, George Couters, who is hiding somewhere here. He's over there, if you want to talk to him. And the third chair is Ben Brown, who's you're going to hear further, is the chair of environmental bioinformatics. And what we are trying to do, actually, is we are led by research. So the core of our activity, it is research-led. We all lead our own research project, but we work in collaboration with a lot of people all across campus. I didn't put the photo of everybody because it would be too many, but we have a lot of collaborative grants, and we work together with people. And the last part, the third pillar of what we do is training, and we try to teach people to become bioinformatician or to use bioinformatics, or to understand what is bioinformatics. Those are the missions that we have. And we do that from actually what was shown earlier by Professor Tim Softly, is that we are talking with a lot of different things all across campus. So there are a lot of efforts ongoing and a lot of investment have been going. So I can start is in the bioinformatics, we have this sequencing at the top, but and we work specifically on cancer because it's a very important part here. But as well, we love population diversity, and we are closely linked with Genomics England. With Professor Gutos, we are linked to the NHS. He's got a shared appointment. We are closely linked with the QE, you may recognize, working with the Institute of Translational Medicine to reach out to people. And if you continue to turn around the wheel, you can see that in environmental bioinformatics, we have actually a link to BIFO, which is if you can't see very well, but it's a big forest where you can do a lot of experiments in it. You have Daphnes to test different types of toxicity and response to environment. And at the top, the Freedom Center that was mentioned earlier, the big investment on how to deal with new types of metabolomic data. So all what we do is pretty much linking with everything all around, and we try to create this center of expertise. So with this picture, you can see where Professor Brown is sitting. So it's my pleasure to introduce him. So he's coming all the way from Berkeley. He's got a joint appointment between Berkeley with the head of the department and Birmingham with the chair of environmental bioinformatics. He will tell you all the beautiful things we can do with this, with one thing in mind, that the center of all what we do is the BEARCloud, is IT. We rely on computer heavily. And we don't need just big power, we need flexibility. So with all that in mind, I will please Ben come in.
Thank you so much, Jean-Baptiste. Really appreciate it. Okay, thanks so much. So what's the crowd like today? How many biologists? How many environmental scientists? Okay, so we'll talk about computers. All right. So I'm a statistician by training, a statistical biologist. My group does a lot of different types of statistical and environmental bioscience. We're very interested in the connection between biological systems and their environments and feedback and systems of that kind. One of my principal charges is to try and develop the area of molecular ecosystems biology. So this is something that was coined in 2008 by Reyes, a fellow named Reyes. And the idea is to start to combine the strengths of cell biology and genetics with ecology and ecosystems biology. So in molecular biology, we're interested in linking information encoded in DNA to processes that occur at the level of cells, at the level of tissues and organ systems, and even at the level of individuals. Particularly in clinical biology, for instance, in human biology, we're very interested in how DNA relates to human phenotype, to the human animal. In ecology, we're interested in a very different task. We're interested in how individual organisms work together to create ecosystems, how populations of individuals interface to create the services those ecosystems provide, like soil nutrient cycling. And a grand challenge for the 21st century is to begin to understand how the information encoded in genomes influences more than just the cell, more than just the individual, more than just even the population, all the way to the ecosystem. Can we begin to go from the nucleotide to the ecology, right? And that is a fundamentally unsolved problem. So it's a field that was conceived in the previous decade, and still progress has been very slow because it is an enormously challenging problem. And part of that challenge is definitely on the computation, which I'll talk about today. So that's the idea, make this thing called molecular ecosystems biology. And the idea is to begin to couple systems on many scales where we can make different kinds of measurements so that we can begin to understand at a molecular level processes that we observe traditionally at only a gross level. So shown there on the far right or left, depending on where you're standing, is a stream mesocosm. That's here at Birmingham, led by Professor Mark Ledger. It's one of the best stream mesocosms on the planet. It's absolutely fantastic. He's got a new mini version of this. It's capable of 48-fold replication. For those of you interested in ecology, replication is always quite a challenge. And then next to that is a terrarium. And then on the other side of the slide is a microbial co-culture. Now, each of these systems has particular advantages. In a mesocosm, what Mark does is he takes water from a nearby stream and flows it into this controlled, built environment. And then he allows it over several weeks and months to self-assemble into a self-sustaining ecosystem, a little mini ecosystem, a lab where he can do experiments. He will perturb things like temperature, chemistry, or even water availability to simulate drought conditions. And that allows him to study in a controlled way processes that otherwise he would just have to observe in nature. And being able to do those perturbation experiments is incredibly valuable. But there are challenges associated with this. It's a complex ecology with many unknowns. Those streams are open to air. You know, spores and microbes are settling in those and taking root. And so there's a lot of uncontrolled variables. The next system over, a terrarium, is simpler. In the stream-fed mesocosm, you don't necessarily know, unless you start sequencing critters, even all of the species that are present, certainly not all of the microbial species that are present. In a terrarium, it's a little bit simpler. You take a defined population that you have put there. You may or may not even control the soil microbiology, so you know exactly the critters that are present. And then in a co-culture, it's simpler than that. You take just a few species of microbe and grow them together and ask what are the metabolic currencies they exchange, how do they live, how do they contribute to life in the petri dish. And what's been missing historically in this collection of systems is something that links the terrarium or mesocosm scale to the co-culture scale. In a co-culture, you have exquisite genetic control over every component. That's not true in a mesocosm or a terrarium. And so one thing that we're working to build are these systems like rhizotrons, where you have a genetically defined system that is completely controllable, molecularly defined ecosystem. And the reason it's been so challenging, the principal reason it's been so challenging to begin to build molecular ecosystems biology is because we have lacked this. In genetics, and the reason molecular cell biology is so successful is because you have this incredibly reproducible process that you can study, and that incredibly reproducible process is development. When an organism develops, it's on a defined trajectory with a defined outcome. Deviation from that defined trajectory is bad, and you can study it. And that has been made genetics an incredible success over the last 100 years and change. What we need now are self-assembling molecularly defined ecologies where we can study defined trajectories through a sequence of state spaces in a self-assembling system, so that I can build a desktop ecology that will be exactly the same whether I set it up in Birmingham or Berkeley. And that is something that we are working on. And this would link in a very nice way, very realistic, with very molecularly incisive models. And then we can make all sorts of measurements, of course, at every scale of this. We can build co-cultures and rhizotrons to explore particular phenomena, particular communities within mesocosms. We can measure gene expression, protein expression, and metabolism in each of these systems and at various levels. And through integrative analysis of these, we can use these systems to refine understanding and progressively make the rhizotron and co-cultures more and more representative of biology that we see in the field. So it's sort of the grand vision. And there's been a lot of progress toward this area. We now have quite a few very powerful enabling technologies. So CRISPR, as I'm sure many of you are aware, that's a genome editing technology that has now made it possible to make very precise edits to the genomes of virtually any critter. It'll probably be a Nobel Prize in the next couple of years. So we can have genetic control over just about any animal that we like, much, much faster than that was previously possible. There are other technologies that allow us to manipulate not individual genes, but individual animals. So there are these glycobinding bead technologies and phage library technologies that are making it possible to do something like remove a single microbe from a community of 10,000 microbes. We have exposure biology. At ecology, the big difference between ecology and genetics. In genetics, if you want to study a gene, you can knock it out and see what the effects are. In ecology, the relevant unit is not necessarily the gene, it's the process. So you're interested in how a particular biogeochemical cycle happens. How does carbon cycling, how does phosphorus cycling work in soil? Well, you want to understand all of the different biological pathways and biochemical pathways that come to bear on that process. And you want to perturb the process and watch what happens and watch how perturbations affect critters. So you need to do something very different than genetics, and that is exposure biology. We design a chemical perturbation to perturb a particular biochemical cycle and then ask how that affects the system. And of course, to bring all of this together, we have high-performance computing, as in BEARCloud. And I'll talk a little bit about how that's enabling some of the work we're doing. There's a lot more we need to develop. We need these model ecologies need to get stronger. We need to develop new types of phylogenomic biology that are better suited to ecology than what we have today. We need to be able to make time series measurements. That's a big one, non-destructive measurements. One big challenge in connecting ecology and molecular biology is dynamical versus snapshot information. In genomics, when you do a sequencing experiment and you say, okay, what are the genes that are expressed? What is the genome? These have historically been snapshots, or if they're done in time courses, very few time points. And in ecology, you're always interested in dynamics. You're interested in systems of partial differential equations that describe relationships in the system. And so moving toward non-destructive omics measurements so that we can gain time series perspectives on natural systems is absolutely critical. And falling prices in omics are making this very, very plausible on the, today. And of course, we need more informative machine learning. And this is one of the central foci of my group, is building statistical and computational tools to interrogate extremely complex systems like mesocosms, like rhizotrons, et cetera. And so I'll tell you a bit about that. Now, when we are going after one of these systems that I've shown you, we generally have several different types of measurements. And we're interested not in the measurements per se, but in the relationships between them. If you want to understand something even as simple as a microbial co-culture, maybe you want to understand the relationship between gene regulation in the individual organisms and their metabolic output. This is very important if you're in biomanufacturing and you're trying to talk a community of microbes into developing jet fuel, for instance. You want to understand what is the relationship between their gene regulation and the output of a particular high-value product. And you want to be able to engineer that. And engineering that means you need a predictive understanding based on variables you can control. So what that comes down to at the end of the day is trying to identify a relatively small set of controls that modulate the process you care about. In modeling terms, that means you're looking for sparse collections of predictive variables that together are highly predictive of sparse sets of states in your output. So sort of illustrated there if you're trying to predict metabolism from gene transcription as in biomanufacturing. This problem is really hard. It's really hard because of something called the curse of dimensionality. There are an enormously large number of genes even in a single organism. And when you talk about a community of many, many organisms, the number of potential predictors becomes extremely, extremely large. And I'll say a little bit more about that. So take just a very, very simple system, just one critter, a human. In the human population, there are approximately 6 million very well mapped genetic variants. So 6 million places where the genome differs between humans. And if for a particular phenotype only a single variant matters, this is a pretty tractable space. So for instance, say you're trying to predict whether or not someone will have some appalling disease, like a Rett syndrome or something. If there's one and only one gene that matters, you're going to do 6 million tests, chi-square test, t-test, whatever. You're going to do some statistical test 6 million times on every variant in the genome, and then ask, if I separate all humans into case and control, they've got the disease, they don't. And then I test, okay, these guys have an A at that point in the genome, these folks have a T. I'm simplifying because we're diploid and this is haploid. But in that very simple case, you do a statistical test and you say, is it significant? Are the folks with this allele enriched for this particular phenotype? That's called GWAS, genome-wide association study. It's very simple, and if one or a few locations in the genome contribute to your phenotype, it's very effective. However, diseases that are Mendelian, meaning that there's one locus in the genome that really controls it, are the vast minority. Most human traits, most traits in any critter, are complex and involve many locations in the genome, not just one. In mice, for instance, there are types of mice that are very sensitive to radiation and types of mice that are extremely robust. There are mutations in the mouse genome that are embryonic lethal in one strain of mice that have absolutely no phenotype in another. And so that's an incredible conditional dependence of the effect of the mutation on genetic background. And that starts to get complicated fast, because if we look at just pairs of variants in the genome, there are 36 trillion pairs of variants. And if we go up to sets of variants 3 at a time, triplets, we've got 2.16 times 10 to the 20th. And that's getting on par with the number of grains of sand on all the beaches on our planet. So that gets to be a pretty big space. And as you go to higher and higher order, this becomes a very intractable problem, and certainly not one you can solve with doing a whole bunch of statistical tests. You have to explore the space in a more principled way. So if we wanted to just understand how genes work 3 at a time for a reasonably complex phenotype, we'd be looking at about 1.5 trillion CPU hours. And even on the most powerful supercomputers in the world, that's still quite intractable. So with the next generation pre-exascale computers that are being built in both Europe and North America right now, and China, like 700 million, a billion CPU hours is reasonable, but 1 trillion is still a hell of a lot. So we've got to figure out how to do things in a smarter way, and that's what we work on developing. And we work on developing lots of different types of machine learning technologies. Just to give you a little bit of background, so who knows anything about machine learning? Machine learning aficionados, right? A couple. Okay, so so machine learning are more sophisticated types of statistical models than we usually have in statistics. One that we're very fond of are called decision trees. Decision trees are very simple objects. You see one pictured there next to the map. That decision tree makes a series of splits of data. So you have a big pile of data coming in, and then you shunt data left or right down the tree, depending on whether or not the data satisfies a particular condition. So this example here is looking at the price of a bushel of apples in California, and then trying to predict the price of a bushel of apples in California based on latitude and longitude of the apple vendor. And the first split slices off Northern California, where apples are quite cheap. And then subsequent splits slice and dice the map of California into a few voxels that begin to approximate the price of apples. Now, any one decision tree gives a pretty coarse representation, which is essentially a histogram representation of data. But when you smooth not just one decision tree, but many, many decision trees together, you can get a very smooth and very precise estimate of a quantity. And this process of smoothing decision trees is known as random forest. It was invented in 2001. It's an enormously powerful form of statistical machine learning. And it's one that we exploit. The challenge with random forest is when you're at a single decision tree, you have a tremendous amount of interpretability. You can look at these latitude and longitude coordinates and very rapidly conclude that apples are expensive in the Bay Area and LA. But when you go to random forest representations, you lose that simple binning, and things get a little bit harder to interpret. So one thing my group works on is trying to interpret more complicated learning machines so that we can develop an engineering level understanding of complex systems. So one algorithm we developed very recently is called iterative random forests. This fits a random forest not just one time, but many times. Each time it fits the forest, we interrogate the fitted object, the learned data representation, and try and understand why that was a good predictor. What was it about the data and about the forest that led to positive predictive power? And by iterating that many times, you make a better and better predictor. And shown there on the right, with the weird tree things and the green lines, is sort of the magic trick behind the ability to do this. At each decision tree, we look at decision paths from root to leaf and ask which variables were made in the splits. In the apple example, we had just 2 variables. It was really simple. In the biosciences, we generally have many, many more than 2. So in GWAS, if you got 6 million variants, then your predictor matrix has dimensions 6 million. So there's 6 million variables to look at, not 2. Big difference. And then you look at a path from root to leaf and you ask which variants or which predictors were important to solve that prediction problem. Now, what we want to find are the set of all subsets of predictors that were important for prediction. But the set of all subsets is still really big. It's called the power set. It has size 2 to the p, where p are the number of predictors. It's still really intractable. But an algorithm was developed in 2015 called random intersection trees that allows you to do importance sampling in the space of all subsets. And so what we do is we collect these decision paths from root to leaf, and then we start randomly intersecting them. and we look for subsets of predictors that occurred many times in many different paths from root to leaf. Those are the important ones. It is a really elegant way of doing all subsets regression, which is something that has historically been completely computationally intractable. Okay, so that allows us to start to explore very high-dimensional spaces. One of the high-dimensional spaces we've looked at in cell biology, just to give you a little more flavor of cell biology, is the one generated by the ENCODE project. So they've done a little over 11,000 experiments genome-wide in human cells. So we now have a very high-dimensional and very powerful biochemical map of the human genome. So when the genome was first sequenced in 2000, we had 2 bits of information per base. We knew whether each base was an A, C, G, or T. And now we have 11,000 and counting tracks of real valued information associated with every base in the human genome. So the genome is a much higher dimensional object now with many, many thousands of bits of information per base. So we like to explore this to study the central dogma of molecular biology, which says that DNA codes for RNA, codes for protein, and we're very interested in how each step in that process is regulated. The regulation of DNA to RNA is extremely well studied. It's called transcriptional regulation. The regulation of translation, how an RNA molecule ends up being translated into protein, is less well studied. ENCODE has generated an enormous amount of data that weighs on the study of this process. And one aspect we're particularly interested in is how chromatin, how the very DNA proximal events lead later to RNA processing decisions. So RNA transcripts, after DNA is transcribed into RNA, the transcript is processed in various ways that ready it for translation. And it's been known for a while that there were a few types of coupling illustrated on the bottom right. There are a few types of coupling of stuff that happens very proximal to the DNA with stuff that happens far away in cell space, as in tens to hundreds of nanometers away, in processing the RNA. And so this sets up a very straightforward sort of prediction problem. We'd like to predict whether or not a given exon in a transcript, whether or not a given bit of RNA is going to be included in the final protein. And that's something that's been studied before, but never in such high-dimensional data. And we can run and have run iterative random forests on this, and we begin to map the surface of RNA processing response as a function of all of the ENCODE data tracks. And we find that certain splicing factors, shown on the middle right, are very important for RNA processing, and that's sort of no surprise. And then shown on the left is the relationship with chromatin, where we see a particular chromatin mark, H36ME3, interacting with other chromatin marks to establish the likelihood surface for splicing. Here's one of the best known interactions on the upper left is the interaction of PTB1 and H3K36ME3. That is a mark on chromatin, so you've got DNA in the nucleus. The DNA has lots of proteins bound to it, and if one protein is modified in a particular way, it creates a docking site for an interaction partner of PTBP1, and that literally creates a bridge, a physical bridge between the DNA and the nascent RNA molecule. And that physical bridge allows the continuous transmission of information between the RNA molecule and the DNA. And that response surface, as far as I know, this is the first time it's been mapped, what that response surface actually looks like. But that was discovered by René Luca in 2010. And what we found using the ENCODE data is that there are 3 other factors, SAFB2, U2AF1, and a HNRNPU, that have a very, very similar response surface. So these may be involved in the same pathway as well. And it's discovered in a completely naive way, right? The ENCODE data was generated essentially without hypothesis, lots and lots of omics data. You run an algorithm like this and you discover some biology. Here is one of my favorite model organisms, the fruit fly Drosophila melanogaster. So the fruit fly was the first animal genetic model organisms developed by Thomas Hunt Morgan in the 1900s, early 1900s. And so it's been a model for a very long time. And as a result, we know a lot about its development. And one thing we're very interested in is mapping not the RNA regulation, but the regulation of the beginning of transcription, transcriptional regulation, and looking at enhancer elements in the early fly. And this is something that's been studied by many, so I won't belabor it. But it's very interesting to see that when you ask what starts a gene being transcribed in the fly, we see a very different type of response surface than we see when we ask how is an RNA processed. So there you see these sort of things that have a big shelf. They're sort of OR-like rules. So an exon is generally included if it has either of those two properties. And here you see much more AND-like rules for the incidence of gene regulation. You need several of these factors to be bound, and if they're bound, you see that nice green spike, and you get the instance of transcription. We've looked at transcriptional control in mouse. In mammalian systems and most animal systems, there's been a dogma for a long time that enhancer elements look a very particular way. They're enriched for H3K27 acetylation, which is a particular type of mark on chromatin, and they're depleted for DNA methylation. So DNA methylation, that's an epigenetic modification to DNA. It's got lots of different roles. But those, so shown on this heat map here, on the right, this is a heat map of all hard enhancers, or all the hard enhancers that have been looked at, about 1,000, 2,000 elements that are active during early embryogenesis in mouse. And the black dots are active enhancers that were provably active in the mouse. And the background, that sort of heat mappy, jet streamy looking thing, those are the enhancers that were tested but turned out not to be real enhancers. They're negative under this assay. And what you see are that lots of the black dots exist there where there's a depletion of negative elements, but then in the bottom right, you've got a bunch of black dots overlapping the core of the jet stream, things that look at a biochemical level as though they should be inactive. And in fact, when we sick our algorithm on this, it actually identifies a saddle-shaped surface, and it says that no, there's actually two types of enhancer elements. They're distinct. There are, of course, lots that are the very classical type that are low methylation and high H3K27 acetylation. But then there's also lots and lots that are hypermethylated with low H3K27 acetylation. And this came as a big surprise, but it's now also been found in zebrafish, so it looks very real, which is quite cool to see that in another model system. So I'm going to tell you just a little bit sort of about a large project that we work on that really does try to start to connect cell biology to ecosystems level processes. And then I'll stop in just a couple minutes so that we can have potentially a few questions. So there's a large consortium called the Microbes to Biomes Consortium. We look at carbon management, food and fuel production, environmental stewardship, and also health. The idea behind the consortium is that we try to do molecular ecosystems biology at the level of microbial communities and the host microbial interface. So think human gut microbiome, think plant root microbiome, et cetera, to try and tackle some fairly large problems. Such as, can we predict the response of Earth's carbon sink to our changing environment? Can we meet increasing global needs for food supply? Can we solve phosphorus production? Things like this. And these are some really tough problems. I don't know if you guys are aware, but one of the most pressing environmental problems is actually not global warming. It's rock phosphorus. Virtually all industrial agriculture requires fertilizing with rock phosphorus, virtually all of which is found in mines underneath Morocco. And we've got about a century left of that stuff. And after that, we will support a population of around 4 billion. That's not great. So that's a really pressing problem. We have to figure out how to cycle phosphorus without constantly needing to add new. And realistically, we've got about 25 years to solve that. So the idea is that we look at genetic variability, microbial community variability, various environmental circumstances, and then try and understand factors that underlie the robustness of various populations or ecosystems. And what I'll just literally, I'm going to fly through just a study that looks at Drosophila melanogaster, and it's a genetic model organism, and its resilience to pesticides in agricultural systems. The same study has been done in mouse, but we won't have time to get to it. So shown up there in the animated bit, those are fruit flies. On the left, those flies are fine. On the right, those flies have been treated with pesticide atrazine, extremely widely used in the US. 33 million Americans drink atrazine-contaminated drinking water on a daily basis. And you see those flies sort of flipping out and spinning around, and you see them highlighted on the image below. That is a unique locomotor phenotype that is induced by even low doses of atrazine. It's not just fruit flies, it's common to other insects. And what we'd like to learn is something about the molecular basis of that phenotype. But also, we've tested flies at some pretty stunningly high doses, and atrazine remarkably does not kill flies. Kills me and you pretty good, but it doesn't kill flies. And so we're also interested in understanding why they get sick but then don't die. So this is the entire transcriptome. So this plot, the big spiky plot on top, that's the linearized genome of Drosophila melanogaster. The red spikes are individual genes. Genes that spike up are being induced. Genes that spike down are being repressed. And what we see when we look at this are essentially a barcode of the toxicant at the level of gene regulation and transcription. And we can interpret some of this. A lot of those genes, thanks to 100 years and change of fly genetics, we know what they do, and we can read something into it. What's somewhat more interesting even than the host transcriptome, because the host transcriptome does not offer any evidence of why they might survive. It offers evidence of why they get sick, but not why they survive. Because the flies only have a couple enzymes that can process atrazine at all, and for the most part they process it into something that is more toxic than atrazine, not less. And so we look at the microbiome, the fly gut microbiome. So we've mapped it now, we know what bacteria is present, and we've looked at what happens to the microbiome over exposure to atrazine and other chemicals too, like paraquat, which is another herbicide. And the microbiome has some really interesting dynamics in response to herbicide. And when we look very closely at the genes that are present, Drosophila actually has a complete community of atrazine metabolizing microbes. And in fact, it turns out that the microbiome is at the root of robustness to atrazine exposure. When we compare flies with a healthy gut microbiome to flies that lack a microbiome, flies that we have made by artificial means germ-free, you see that the flies that have a healthy microbiome can survive. This is a very survival curve for a very high dose of atrazine. And flies that lack a microbiome die. And what we're working on right now is the rescue experiment. And we do not have a plot yet, but what it looks like, we can actually rescue a fly that's been exposed by giving it the correct microbes, which is quite cool, because if we can start to do rescues for particular toxicants with particular microbiomes, you can start to treat things like 33 million Americans exposed to atrazine with probiotics. It'd be very nice to treat disease with yogurt. So where we are right now with machine learning is we can find cute things like I've showed you. We can find relationships between particular molecular processes and particular outcomes. But where we need to be, especially for the ecological dynamics, is in the setting of dynamical systems. We need to be able to infer in a very automated way, not just those cute static pictures I showed you and those cute rotating pictures. Those things need to be dynamical. We need to understand how these systems evolve in time. And that's plausible over the next several years that we could get there from where we are. And so just a thank you to the entire Health M2B team. Thank you very much for your attention, and I'll take any questions.
Thank you, Ben. Thank you. Perhaps just one question. Keep us quite tight to time. I suppose my question is, how much more computer power do you need?
As much as you can give us. The more compute power, the more we can explore. With machine learning and with this iterative random forest and a host of related algorithms, what we've done is begin to decouple the cost of computation from the order of detectable interaction. And that's really important, because it gets rid of the need for things like forward regression that have historically made it impossible to detect episthetic relationships in things like GWAS, or really complex relationships. So that's overcome, and that's fantastic. But we also need much more sophisticated models and dynamical models. And to fit these things right now, it takes several 100,000 to 1 million plus CPU hours per model. And that's for a static snapshot. And now we have to go dynamic into video mode. And so saying that, we're looking at still, hundreds of millions of CPU hours, if not more. So quite a bit of compute power.
Okay. Okay, guys, we're going to drop for a while.